We are about to redesign the human body. Not metaphorically, not in some distant science fiction future, right now. With tools that already exist, in laboratories that are already open, by scientists who are already doing it. And almost nobody outside those laboratories is having the conversation that needs to be had about what that means. This video is that conversation. In 2012, two scientists, Jennifer Doudna and Emmanuel Charpentier, published a paper describing a molecular tool that could edit DNA with a precision that had never before been possible. The tool was called CRISPR-Cas9. They won the Nobel Prize in Chemistry in 2020, not because the science was elegant, though it is, but because what they had done was hand humanity a pair of scissors that could cut the instruction manual of life at any point they chose, and then rewrite it. CRISPR works by using a guide molecule to locate a specific sequence of DNA out of the 3 billion base pairs in the human genome and cut it with extraordinary precision. The cell then repairs the cut, and if you provide a template during that repair process, the cell will incorporate the new sequence you wanted instead of the old one. Find, cut, replace. It sounds simple. It is simple relative to everything that came before it, which is exactly what makes it so significant. Before CRISPR, genetic editing was possible but extraordinarily difficult, slow and expensive, the kind of thing that required years of work by specialist teams with budgets most institutions couldn't sustain. CRISPR changed that almost overnight. Within a few years of the original paper, labs around the world were using it. The cost dropped by orders of magnitude. The speed increased by orders of magnitude. High school biology students were conducting CRISPR experiments. This is the moment in any technology's history when things start moving faster than society can track. What can CRISPR already do? In medicine, the early results are extraordinary. Sickle cell disease, a devastating inherited condition that causes episodes of severe pain, organ damage, and shortened life expectancy, has been treated with CRISPR in clinical trials with results that look close to curative. Patients who had spent their lives managing a condition that had no solution are in some cases functionally cured. Certain forms of blindness caused by a single genetic mutation have been treated with CRISPR injected directly into the eye. These are not theoretical outcomes. These are people, real patients, before and after. Cancer treatments using CRISPR-modified immune cells are in clinical trials with promising early results, and this is CRISPR in its infancy, used carefully. On somatic cells, cells in the body that are not passed on to children, the changes affect only the individual being treated. That distinction matters enormously because there is another kind of edit. In November 2018, a Chinese scientist named Jiankui announced that he had used CRISPR to edit human embryos that were then implanted and born as children. Twin girls, Lulu and Nana. The edit he made was intended to disable a gene called CCR5, which produces a protein that the HIV virus uses to enter cells. The stated goal was to make the children resistant to HIV. The scientific community reacted with immediate and almost universal condemnation, not because the goal was unreasonable, but because Jiankui had crossed a line that the scientific community had explicitly agreed not to cross without far more research, far more ethical deliberation, and far more regulatory oversight. He had made germline edits, changes to the DNA of embryos that would be present in every cell of the resulting children, and that would be passed on to their children and their children's children, permanently and heritability. He had, without adequate consent or oversight, altered the human germline for the first time in history. He was sentenced to 3 years in prison by Chinese authorities. The children, now school age, are being monitored. We do not know the full consequences of what was done to them, but the line had been crossed, and once a line like that is crossed, the question stops being whether it will happen again and starts being when and by whom and under what circumstances. Here is the ethical landscape we are now navigating. The therapeutic case for CRISPR is genuinely compelling. Diseases caused by single gene mutations, and there are thousands of them, are in principle curable with CRISPR. The suffering that could be prevented is real and significant. The argument for moving forward carefully but decisively with somatic gene therapy is strong. The enhancement case is where it gets complicated. Once you can edit genes to remove disease, the question of where disease ends and difference begins becomes urgent. Is deafness a disease? Some deaf communities would argue strongly that it is not, that it is a different way of being human with its own culture and identity. Is short stature a disease? Is a predisposition to depression? Is a lower than average IQ? And beyond the disease question, if you can remove genetic disadvantages, can you add genetic advantages? Higher intelligence, greater physical capability, reduced susceptibility to aging, enhanced memory. These are not science fiction questions. They are questions that CRISPR has made practical within a time frame that is much shorter than most people realize, and they are questions we are almost completely unprepared to answer as a society. The distribution question is the one that keeps me up at night. If genetic enhancement becomes possible and safe, who gets access? The history of medical technology is the history of unequal access. New treatments begin expensive and exclusive. They become cheaper and more widespread over time. But the gap between early access for the wealthy and broad access for everyone else is measured in decades. Decades in which the children of wealthy parents could be enhanced, cognitively, physically, medically, while the children of everyone else were not. We already live in a world where the advantages of wealth compound across generations. Now imagine adding genetic advantage to that compound interest. The children of the wealthy would not just have better schools, better nutrition, better healthcare. They would have better genes, deliberately chosen, professionally edited, passed on to their own children. That is a world of two biological classes of human, enhanced and unenhanced, divided not by accident of birth, but by parental bank balance. We have not begun to seriously grapple with what that means. And the technology is not waiting for us to catch up. There is one more dimension worth sitting with. The countries moving fastest in this space are not necessarily the countries most concerned with the ethical questions. Regulatory frameworks for genetic editing vary enormously across the world. What is prohibited in one country is permitted in another. What requires years of ethical review in one jurisdiction can be conducted with minimal oversight in another. This creates a race dynamic that is deeply uncomfortable. If country A prohibits germline editing on ethical grounds and country B does not, the pressure on country A to revise its position becomes enormous. The argument that your children should have access to the same advantages as children born elsewhere is not an easy argument to resist. We have seen this dynamic before. It does not tend to resolve in favour of caution. So where does that leave us? CRISPR is real. The therapeutic applications are already saving lives and will save many more. That is genuinely wonderful and should not be lost in the anxiety about what comes next. But what comes next is coming. The germline editing question is not closed. It is open, contested, and moving faster than our ethical frameworks can track. The decisions being made now, in laboratories, in regulatory bodies, in government ministries, in boardrooms, will shape the genetic future of the human species, permanently. Heritability means forever, and most of those decisions are being made by a very small number of people, largely without the rest of us in the room. That needs to change, because this is not a decision for scientists alone. It is not a decision for governments alone. It is a decision for all of us, about what kind of human beings we want our descendants to be, about what advantages we consider acceptable to engineer and what we consider too dangerous to touch, about whether the human germline belongs to humanity or to whoever can afford to edit it first. These are the hardest questions, and we are running out of time to have them carefully. Next time, another question that doesn't have a comfortable answer. See you then. If this gave you something to think about, hit like. It helps more people find these ideas. Subscribe for a new video every week. The future isn't going to wait. Neither should we.